Great. So thanks so much for the invitation. So as Mario said, I'm based at Weill Cornell currently, but I'm Canadian, actually in Canada at the moment, in Vancouver. Going to be visiting Sabrina Brume's lab after this. So I wanted to talk to you today about something my lab's been interested in for a while, and this is this idea that one of the ways that HIV-infected cells may persist on therapy is that the cells that harbor the reservoir are selected for those that are intrinsically hard for the immune system to kill. So I'll get into a bit more about what I know about that. But just by way of a little bit of background, we know that the CD8 T cell response is effective in partially controlling HIV replication in most people, and in some people that we call elite controllers can actually suppress viral replication to undetectable levels. So this is just a cartoon. The first clue to suggest that CD8 T cells are playing a role in viral control is that these responses emerge at the same time that viral load comes down from a peak to this lower set point. And there's many other lines of evidence that have emerged since, and I just wanted to show a little bit of data from our group as well that'll tie into some later parts of the talk, showing that CD8 T cells can also control HIV replication in the humanized mouse model. So this is a model where we engraft mice with CD4 cells from a person living with HIV, someone who is an elite controller, and then either we give autologous CD8 T cells, we don't do anything to manipulate them, we just give them directly, or we maintain them as no CD8 controls. And you can see just this addition of CD8 T cells is sufficient to bring the viral load down by a few orders of magnitude here, even though control is not 100% complete. So we know that CD8 T cells can have in vivo antiviral activity. So the question is, once we are able to suppress viral replication to undetectable levels with antiretroviral drugs, why don't CD8 T cells finish the job that they started, eliminate those last infected cells? And we know that generally this doesn't happen because if you stop antiretroviral therapy, of course, viral loads rebound. So what is the limitation here that stops these CD8 T cells from finishing the job? So the classical thinking here is kind of binary and a little bit simplistic, I would say, in that the standard model is that the infected cells completely hide from the immune system. They're in a latent state. They don't transcribe viral transcripts. They don't produce proteins. And so the immune system simply doesn't see them. And if they do become activated, then those cells will go on to die. So this is, I think, how the reservoir has been thought of for a long time. And this is no doubt an important part of how HIV persists. So latency is important. But what I'm going to talk about a bit today is this idea that some of these cells that are producing virus nonetheless seem to be able to persist in people. And so latency doesn't seem to be the full story, as I'll elaborate on in a minute. So moving from this idea of a completely latent reservoir to cells that are producing virus and die, the field has really learned a lot over the past few years in developing a more nuanced understanding of how HIV persists. I think it's really been an exhilarating time to be in the field and see this developing so quickly. So first of all, we know that the reason that some HIV DNA persists in people on therapy is that a lot of it, 98% or so of the HIV DNA you find in people on therapy are these defective proviruses. Some of these can produce antigens, but some of them also are completely dead from a protein expression perspective. So if we want to measure a change in the reservoir, we need to focus on the intact proviruses. And there's ways that are developed now to kind of look at this. The second issue is that it's extremely important to think of where the HIV provirus is integrated into the genome of a cell. And if you actually look very carefully over time in people who are controlling HIV, or even people who've just been on therapy for 20 years, what you begin to see is that the proviruses that remain are those that are inserted into parts of the genome that are not amenable to transcription. So they're in centromeric regions, they're in gene deserts. And so those proviruses might stick around, but are perhaps not expressed. So that's another kind of aspect of how latency might be selected for. We also know now that infected cells undergo clonal expansion. So infected cells, even if the virus isn't replicating because of of antiretroviral therapy, the cell itself can divide. And this is actually a very dynamic process. Some clones of infected cells wax and others wane is kind of the terminology that the field has started to use. And the processes that drive those dynamics are not completely understood, and an important part of understanding how we can push some of these clones to die off. And then finally, the concept that I'm going to talk about today is this idea that some of these cells that are infected might get recognized by the immune system and if not efficiently killed off despite that recognition. So to focus in on that idea of CTL resistance, I want to take a bit of a step back and talk about how we were thinking about the interaction or the lack of interaction between CTL and the reservoir a few years ago when I wrote this review with Bruce Walker. And we proposed at the time that there was these 4 barriers that were preventing these CTL from targeting and killing these cells. First, of course, sequence diversity and immune escape. If the virus in the reservoir has been selected for those proviruses that aren't recognized by the CTL, that's going to be a barrier to killing it off. The HIV-specific T cell response becomes progressively dysfunctional, losing proliferative capacity most of, but also cytotoxic capacity. There's an issue of compartmentalization. So for example, there's an important viral reservoir that's present in lymph node follicles where cytotoxic T cells don't really enter. And there's this issue of HIV latency where some infected cells aren't expressing protein and aren't getting detected by the immune system. But we started to ask ourselves a few years ago, is this actually a complete and accurate model of the barriers preventing CTL from killing the reservoir? And 3 main points made us think of this. First of all, a lot of clinical trials had tested this kick and kill concept, and some had shown increases in viral RNA, but no decreases in the reservoir. So this may just be because the LRAs aren't strong enough, the immune responses aren't strong enough, but we thought it was also a good time to take stock of this overall model and see if we might be missing something. Second of all, evidence from our group and others have shown that there's actually pretty substantive in vivo recognition of antigen by HIV-infected T cells. So if you look at the HIV-specific T cell response, it not only persists for years or decades in people on long-term therapy, T cells, in particular those that recognize early gene products like NEF, maintain this short-term granzyme B release when they're stimulated. And that's really a hallmark of T cells that have recently seen their antigens, so effector T cells. So if you ask the T cells if they're seeing the reservoir, essentially in people on long-term therapy, the answer is to some extent, at least, yes. And third, we essentially tried to achieve this kick and kill approach to reducing the reservoir ex vivo, take CD4 cells from people on long-term therapy, hit them with strong latency reversing agents, come in with cytotoxic T cell clones that circumvent all of these issues. So we take clones that we know target non-escaped epitopes that are highly functional because they're exposed to all the cytokines they want. There's no issue of compartmentalization in vitro, and we're using strong latency reversing agents. However, the outcome of this, which was published a few years ago, is that when the dust settled on these assays and we measured how much reservoir was left by viral outgrowth assays, we hadn't really moved the needle. And if we took the viruses that grew out from the viral outgrowth assays that we use to measure the reservoir and infect new cells, our same cytotoxic T cell clones could kill those newly infected cells. So it made us start to think about these actual original cells that are harboring the reservoir in people on long-term therapy that have survived for many years. Is there something about these cells themselves that might make it hard for cytotoxic T cells to kill them? And so thinking about this additional barrier, the need to identify and overcome mechanisms of CTL resistance. And I'm going to show some data now from a recently published paper from our group showing that BCL2 is an important factor in this that might be an important therapeutic target. And some groups are exploring BCL2 antagonists in the context of HIV. And then I'll talk about some unpublished, more recent leads of other mechanisms that might make cells resistant to cytotoxic T cells. So first of all, what might be the origin of CTL resistance in the reservoir? And an important distinction is that I'm not saying that HIV-infected CD4 cells in general are resistant to being killed. Certainly, if you take primary CD4 cells, infect them in vitro, and try to kill them with a cytotoxic T cell clone, you kill 90% of them. but you never really kill 100% of them. There's always this population that survives. And so the idea is that a small fraction of infected cells might have properties that make them hard to be killed. And if that's the case, then it stands to reason that those cells would preferentially go on to form this reservoir that persists in people who continue to have this CD8 T cell pressure. And so we propose a few different aspects here. So first of all, the infecting virus could drive this effect by having CTL escape or superior NF function. That's well known. So that's not kind of a new approach that we're focusing on. There might be just differences between host cells. We know naive cells are hard to kill, memory cells are easy to kill. What other aspects of heterogeneity within CD4 cells determine how susceptible they are to being killed by a cytotoxic T cell? And HIV, of course, samples a huge landscape of CD4 cells within a person living with HIV. So there's a lot of heterogeneity that can be selected by the immune response. And finally, this idea that there could be a unique aspect of actually how the virus and the host cell interface with each other. So does the integration site, for example, change in some way the function of this cell that makes it intrinsically more resistant to being killed? So for the purposes of today's talk and with the time allotment that I have today, I'm just going to focus on this first possibility, which is that there's just differences between how susceptible CD4 cells are to being killed by cytotoxic T cells, and that HIV can happen to land in some cells that are relatively resistant to being killed. So to ask this question initially, we took central memory T cells, primary CD4 cells, which are an important aspect of the HIV reservoir, pulsed them with peptide, mixed them with unpulsed cells, tried to kill them with cytotoxic T cell, and then just performed transcriptional profiling on what we call the survivors here. So this small population of peptide pulse cells that wasn't killed despite an excess of potent cytotoxic T cell effectors. And what we found from our transcriptional profiling is there was a lot of differentially expressed genes. There's actually a lot that we still need to go through from these published data, but there were a lot of pathways that were converging on BCL2 as a kind of central regulator of this process. As we can see here that BCL2 is overexpressed in the cells, the survivors of these killing assays. Caspase-2 is also underexpressed. This is an important effector molecule in the process of cell death, etc. But we decided really to focus here first on BCL2 as something that might be overexpressed in cells that are relatively hard for the immune system to kill. So how might this work? So the cancer immunology field is way ahead of us in this concept. It's well known that certain tumors can be selected for those that are intrinsically hard for the immune system to kill. And so this is a review from that field. And this is a target cell. So the target cell will receive an inducement to die, either from perforin granzyme or one of these death receptor ligands. And normally that's kind of where our thinking stops. If you have a target cell, it's expressing antigen, it receives a cytotoxic hit, we think that that cell is going to die. But what the cancer field can teach us is there's a lot of regulation that happens downstream of this. And there's negative regulators of this death process that can prevent that cell from dying. And one of these negative regulators is BCL2, which can sequester some of these pro-apoptotic proteins that are activated by this cytotoxic T cell inducement. So there seemed to be good rationale for this hit that we had found. This was in a peptide pulse setting. So we wanted to ask, does this actually have relevance to the HIV reservoir that is in people living with HIV? So we used an assay developed in Montreal by Nicolas Chamont's lab called HIV Flow, which lets you detect and phenotype these very rare cells that are expressing HIV after you reactivate them in CD4 cells from people on therapy. And we just measured BCL2 in these cells. And even though there's some variation across these different individuals, what you can see is that consistently the HIV-infected cells express more BCL2 than the uninfected cells in these people on long-term therapy. And we don't really see this in cells from ART-naive individuals. So starting to get at this idea that it's not that HIV-infected cells in general overexpress BCL2, it's that the infected cells that we find after we look after years of therapy, these real survivor cells, show that there's this population is overexpressing this pro-survival factor. We can also do this another way. This is with reactivation. We can simply take cells ex vivo and flow sort them on BCL2 high versus BCL2 low populations and look at the frequency of reservoir harboring cells, these intact proviruses within these populations. And we see it's really this BCL2 high population that contains most of the reservoir. So most importantly, with this aspect of the story, we went back to our original ex vivo assay where we're combining latency reversing agents, cytotoxic T cell clones, and initially we're not seeing reductions in the reservoir. So this is a viral outgrowth assay by QVOA. And we added to this a BCL2 antagonist called ABT199. And this antagonist with latency reversal alone was also not enough to decrease the reservoir. But finally, after actually quite a few years of trying, this triple combination of this latency reversing agent, the HIV-specific T cell clone, and this BCL2 antagonist was able to unlock this reduction in HIV reservoirs that we hadn't seen with the dual combinations. So that's encouraging, I think, but there's also some drawbacks. First of all, the depletion is not complete. Second of all, there's quite a bit of bystander toxicity with this molecule. So we were careful to try to isolate the impact on the reservoir separate from the impact on overall cell viability. Nonetheless, it's a complicated molecule to work with, and the corresponding drug does have a side effect profile that makes it, I think, kind of marginal in terms of potential for testing in people living with HIV. So there is a licensed drug that can target this pathway. It's called venetoclax, and the mechanism of action is shown here. So there are studies going forward, I know, to test this in non-human primate models. And I think it's a question for the clinicians and clinical trialists, whether or not there's good rationale to test this in the setting of otherwise healthy people on long-term therapy. In the meantime, I want to pivot away from BCL2 into a new hit that we've recently found. These are, again, transcriptional profiles of cells that survive CTL killing from 2 different donors. And what we found is that we have consistent overexpression of this gene called EZH2 in the cells that survive killing. So what is EZH2? It's the active unit of something called the polycomb repressive complex 2. So this acts to methylate histones and shut down transcription of certain genes. And one of the genes that this can actually repress is MHC-I, which is important, which is the key protein in presenting viral antigen to cytotoxic T cells. So we hypothesized that this was acting by regulating levels of surface MHC-I. And there are also EZH2 inhibitor drugs that are available, including a licensed drug that I'll talk about in a moment. So we were able to take CD4 cells from these infection cultures, treat them with this EZH2 inhibitor. What you see is that HIV-infected cells downregulate surface MHC-I a bit due to the actions of NEF. And we still see that if we treat with the EZH2 inhibitor. However, the overall population has shifted upwards once we inhibit EZH2. And so now, even with the downregulation in the GAG positive cells, the level of expression of MHC-I becomes more similar to the uninfected cells in the absence of drug. So we think this is another potential strategy to enhance the susceptibility of the reservoir to cytotoxic T cell killing. So in vitro, this certainly works. So here's an infection culture. This is the percent of infected cells that we have without CTL. EZH2 inhibition on its own does not have an impact. If we add cytotoxic T cells only, we get a reduction in the percent of infected cells, but we get substantially greater reduction if we also inhibit this EZH2, which we know is increasing surface levels of MHC-I. So we, of course, now need to take this to our ex vivo reservoir, or kick and kill assay, and ask whether this actually sensitizes the reservoir harboring cells to elimination. So those studies are in progress. But I wanted to emphasize something else on this front. So Mario talked about the opportunities to cross-fertilize with the cancer field and to learn from each other different ways to enhance immunotherapies, for example. So it was really rewarding that we did studies with these EZH2 inhibitors in a B-cell lymphoma line. We use this just because it's an easy source of autologous antigen-expressing cells that we can generate from people with HIV. And we find that if we knock out this EZH2 gene, we actually get better killing of this B-cell lymphoma line, consistent with our results with HIV-infected cells. And this experiment really coincided with this publication from John Udell's lab, which identified EZH2 as well as this other gene as key negative regulators of antigen presentation in B-cell lymphoma as well. So this was an opportunity, I think, where the cancer field could have also learned from our results. And so we hope that a lot of the other hits that are coming up in our screens will be things that might have relevance in that setting as well. And we can, of course, benefit from them as well, because there's also an EZH2 inhibitor called tezometastat, which is licensed for follicular lymphoma. And so we probably need to do some humanized mouse models to have some preclinical in vivo data, but we think there may be some potential to test this as an additional approach in HIV. So to summarize where we're at with this project, CD4 T cells harboring intact proviruses are relatively resistant to killing by CTL ex vivo. We have published results and also some new, more direct evidence that I didn't have time to go through on this, but I think that this conclusion is pretty well supported. In vitro, we have productively infected cells that survive killing, showing a distinct and actually very complex transcriptional signature. So we've actually been just kind of picking relatively low-hanging fruits so far, but there's a lot to mine in these data sets to look for hits with, to look for new therapeutic directions. So there's a lot of hits that come up strongly in this data that we don't really know how they're influencing CTL killing at this point. A lot of things involved in cytoskeletal regulation, for example. So maybe it's the physical properties of the target cells. And so we're taking a two-pronged approach, validating individual targets as we've done with BCL2, because we want to get some things moving down the potential preclinical and clinical pipeline in the short term. And we think a much longer term goal is to try to work towards a more comprehensive understanding of the pathways and upstream regulators that are governing this process. Just on the last slide, how might we think about immune resistance and how this contributes to persistence in relation to HIV latency? And when I present this story, I often hear kind of a negative interpretation or discouraging interpretation. So even if we overcome latency, we still have to deal with resistance. Isn't this a problem? And I would encourage thinking of it more in this way. So we know that there are cells that continue to transcribe and express HIV in people on long-term therapy, in particular in tissues. And so we would propose that this more active reservoir, it stands to reason, would be more selected for CTL resistance and more susceptible to these types of drugs that might overcome this resistance. So we think it's reasonable to suggest that we can make a dent in this reservoir even without latency reversing agents. And then probably combinations with LRAs are needed as we start to shift towards this end of the spectrum to cells that are in kind of deeper states of latency. So I think I'm right on time. So with that, I'll wrap up and thank Louise Layer, who's been leading the intrinsic resistance project overall. Andrea Grammatica did the work with EZH2. Thank our lab members as well as our clinical collaborators, especially Erica and Colin at the Maple Leaf Medical Clinic and the study participants that they recruited. And thank you so much for your attention. Happy to take any questions.